Greetings to everyone. Today we will continue with the chapter number 15, that is plant growth and development. So yesterday we looked upon the plant growth attributes, that how to measure growth, what are the parameters with which we measure growth. We compared growth and development. We studied about phases of growth, what are the factors affecting growth. Now, today the main topic of this discussion will be growth regulators. Obviously, growth is very good, but uncontrolled growth is dangerous, both for plants and animals. In case of animals, uncontrolled growth means there will be cancer. In case of plants also, there is possibility of uncontrolled growth which may lead for tumor. Plants also have gall cells or tumor cells. And body of a plant, animal, whichever organism is moving in a specific rhythm. So something, anything which is coming in between that rhythm is going to break the rhythm. So obviously uncontrolled growth is not expected. Now, how these plant hormones or phytohormones are very efficient in maintaining the growth. So plant growth regulators or plant hormones are very simple molecules, and over the period of time, one by one, they were discovered by many people, and they can be broadly classified into two classes. One are plant growth promoters and other one is plant growth inhibitors. So promoters, as their name itself is suggesting, will promote growth. That means they will promote cell division, they will promote enlargement, that is elongation of the cell. They will promote the trophism, like we were discussing about phototrophism. So in phototrophism, the plant moves towards the light, and it's a trophic movement where the movement is promoted by the plant in the case where growth is responsible for twisting of the stem. So the hormones which are promoting them are auxin, gibberellins, and cytokines. But obviously, there should be antagonistic hormones. While we were studying the chapter number 22, we observed that in animal hormones also, if insulin is doing a work, there should be some opposite functional, that is glucagon. Here also, along with the plant growth promoter, we must have inhibitors. So they are promoting inhibition. How? By putting a plant into dormant stage. Now, what is this dormancy? Inactive stage. They do things minimum for survival, but they don't go for reproduction and other process. Okay, they are just, obviously they are not metabolically inert, but they are not actively living. Okay, apart from that, abscessization is there. Abscessization or abscessization is something where the leaf will fall down, the fruit will fall down. This is a way for plant to excrete the substance. This is a way for plants to get rid of the unwanted material they have. Because like us, we are having urea and uric acid, or some other animals are having excretory substance to get rid of. Plants also are having reactions in them. It's not the case that they don't have metabolism. So obviously, during this reaction, they are also producing certain things which are not wanted. They may be toxic for the plants. So they get rid of those substances by sending them to the part of the plant where they can be deposited. And if that part of plant is falling out of the plant or getting detached, plant is getting rid of that part. Apart from that, a plant, whenever is like in a stress condition, for example, if plant is having a wound, okay, or plant is having a stress like water stress, or plant is having like hot climate, or it is infested by some kind of insect or pest. In such condition also, growth inhibitors show their colors, and they also kind of control the performance of plant. So the growth inhibitor hormones are abscisic acid and ethylene. Okay, ethylene actually can be in some cases growth promoting, but mostly it is inhibiting because it helps in ripening or aging of plant part. So that's why we are broadly categorizing it itself into a plant growth inhibitors. So we will start one by one. Um, First, we will talk about auxin. Auxin is not a single hormone. First of all, let me tell you, many people have misconception. Auxins are group of hormones. Okay, so actually Charles Darwin and his son Francis Darwin, they were working on the coleoptile of canary grass. So can anyone tell me what is coleoptile? Covering of in case of, very good. So that is the covering of plumule in monocot, that is canary grass. They observed that if you are giving them a light source, the coleoptile will have a tendency to move towards the light source. In this diagram, you can see that 4 samples are there, A, B, C, and D. A, B, C, and D. Now you can see the D case where it is moving towards the light. The coleoptile portion marked here is in as green. Okay. Now, auxin was first isolated from the tip of oats seedling by F.W. Wendt. But let me tell you, in our urine also small amount of auxin is there. Although they are not working here as a growth regulator, but as I said, a type of auxin is available in our urine also. So, yes. First was urine, and then it was in plant source, it was oats. So, basically, auxin is a hormone, plant hormone, which is always synthesized in the tip, that is either root tip or shoot tip. The way we are having specialized gland for secretion of hormones, plants do not have such kind of thing. They have group of cells which are able to synthesize the hormone, and in case of auxin, these cells are located at the root tip or the shoot tip. Okay. So there can be various types of auxin. Some are natural. That means they are found in different plants that is available in nature. These are IA, indolactic acid, indole butyric acid, because they are isolated from plant. But over the time, people thought of creating such molecules by modifying some structure of IAA and IBA, and they were successfully able to create artificial auxin, which is naphthalene acetic acid and 2,4-D, that is 2,4-dichlorophenoxyacetic acid. One name is not mentioned here, but I wish to mention it is NOA, naphthoxyacetic acid. That is also a man-made, that is synthetic auxin. Okay, NOA, naphthoxyacetic acid. So these are group of auxins, but let us focus more on what they do. So the function of auxin is, first of all, obviously elongation of plant via means of promotion of cell division. So they obviously promote the growth of plant. Now, apart from that, we know that plant hormones, although naturally produced in plants, we always try to use them for our benefit. So obviously, these plant hormones, when were extensively studied by scientists and their role was clear to them, we started using them for two purposes. One, for agricultural purpose. Second is for tissue culture. In case of grade 12 students, you know what tissue culture is. We discussed in detail. In case of grade 11 students, I wish to talk about it. Some part of the plant is collected, sterilized properly with a specific measure, and then they are kept into a test tube. उनको एक टेस्ट ट्यूब के अंदर रख दिया जाता है in sterilized condition. ऐसा नहीं कि एक प्लांट को हाथ से ऐसे उखाड़ के टेस्ट ट्यूब के अंदर डाल दिया. There is a specific instrument which doesn't allow infection, which is called laminar, जिसके अंदर बैठ के inoculation किया जाता है, and the inoculation or addition of the plant into the medium is done in sterilized condition, ताकि कोई भी उस मीडियम में सिर्फ और सिर्फ कौन ग्रो करे, प्लांट, no other one. Okay, so in that medium, which is called nutrient medium, we can add the plant hormone as per our requirement. We know that plants are totipotent. Yesterday we discussed, है ना? So it will be depending on us or decided by us what I want from this plant. Even if I am taking a piece of leaf, I can get a shoot from that, root from that. So that is the way auxin is very good in promotion of root. Especially IAA and IEA is used in adequate amount. It is promoting excellent rooting. So auxin can promote rooting into the stem cutting. How and where? So how it is promoting the stem cutting? What we do, we take a cutting of stem. For example, this is very commonly used for rose, mangoes. Okay, what we do, we took a piece of stem, dip them into auxin, and then keep them in the like implant them into the soil. Naturally, even if we are implanting, there is formation of root, but auxin will make sure that this rooting will be faster and more efficient. So what will happen in normal condition without auxin, if you are keeping it, in that case out of 10 maybe 4 will die, but if you are putting auxin, 9 may have the rooting and they may survive. Okay, so this is the first role of auxin, that it can promote rooting. Second role is, in some specific plant it is responsible to promote flowering. Okay, so third thing is, it is making plant fresh. That means it doesn't allow the fruit and leaf fall at early stage. So there are some malnutrition in plants. We will see in chapter number 12 that every element is required in adequate amount in plants. All of you must be knowing that gardening is a very interesting thing, and you can go for gardening. It is very good for environment as well as for yourself. It says that it may give the there is a quotation, if you want to be happy for one day, do this. If you want to be happy for 2 days, do this. There is a quotation. I will sometime take it out and let you know. So it says that gardening can give you peace of mind, and let me tell you, these hormones are nowadays used extensively in gardening. So there are some cases where if there is any specific kind of nutrition deficiency in that specific situation, what will happen? Plants will have early leaf fall or early fruit fall. It is very dangerous because without leaf, plants cannot have a healthy life, isn't it? So in such situations, what will happen is auxin can be sprayed. It is a very good thing to stop the premature leaf or fruit fall. Okay, but the funny thing is, it is promoting abscessization on the older leaves or fruit. Now, what is this abscessization? So, what it does is leaf or fruits which are actually attached to the stem at the base of that leaf or fruit. That means the part where petiole of leaf or the stalk of fruit is attached to the stem. There is deposition of abscisic acid. This presence of abscisic acid will make sure that the hold of that stalk with the stem will get weak. वहां के cells क्या हो जाते हैं? Degenerate हो जाते हैं. So what will happen? If there is small wind or even slight vibration, the leaf or fruit will fall. So this is promoted by auxin. So if it is a good leaf, it will prevent it to fall. And if it is an old leaf, the same auxin will promote the falling of the leaf. Next important thing is, it is very, very useful to promote the parthenocarpy. Grade 12 students know what is parthenocarpy. Can any one of you? Bolo Jia. How many Jias are there? One only, I guess. Speak, Jia. Without formation of fruit without the fertilization. All of you require something, a shock come out of. Anyway, so it is the situation when fruit is produced but fertilization doesn't occur. So what will happen? There will be no seed formation. Fruit is seedless, and trust me, nowadays they are on demand. Although they are not very good from the point of view of biology, because that fruit is of no use. It cannot reproduce. Okay, but seeds of tomatoes are avoided by many people. Like if you are having uric acid, I never know, any such thing is not reported in medical science. In such cases, seedless tomatoes can be produced by this method. Even for apple, this is used. Auxin is used. Next is a very good herbicide, that is 2,4-D, that is 2,4-dichlorophenoxyacetic acid. It is very, very important to kill the weeds. In a field, when we are going for agriculture, it is our sole interest is to get the harvest. But if in the same field along with my plants, there is like presence of weeds, that is really unfortunate, because every manure you are giving, the place you are keeping for your plant, sunlight, the water you are giving, the care you are giving, everything will be shared with the weed, and you will not get any harvest from it. So we should not recommend weed. So it is very good to kill the weed. Why? Because what happens is, the monocot plants, for example, in case of main rice crop or wheat or jawar bajri, the what I can say, grains we cultivate in land are mostly all of them are monocot, isn't it? Maize, sorghum, barley, jawar, bajra, rice, all of them are monocot. So they have what? They have small elongated leaves. Monocots are having parallel venation, if you can remember. But the weeds generally found in that area are dicot and having broad. So what happens if you are spraying 2,4-D? Monocots are not affected by these 2,4-D because of their leaf structure. Whereas dicot with dorsiventral leaf are broadly affected by this herbicide and then they die. So you don't have to go to manually to the agricultural field and uh, uproot each and every weed. Okay. By spraying Oxine, you can get rid of the weeds. Apart from that, it is helping the differentiation process and control the cell division. So that means it is promoting always the growth of root. So all of you know that it is helpful for promotion of phototrophism. So can anyone tell me how Oxine promotes phototrophism? So I will describe, no problem. What happens is in a plant, consider this is the plant. Okay. Now at this side there is presence of sun. So what will happen, beta? Sunlight is coming to this area. So Oxine is having a specific property that it hates light. It gets always stored into the portion which is in the opposite part of the line. That means it will be always stored in the shady part of the line. So what will happen? All Oxine will be stored here at the other side of the stem. So what will happen? So the growth at this part will be more. So the stem will bend like this, will bend towards the. It will bend. It will bend towards the light. So this is how Oxine promotes phototrophism. I want you people to take a note of it. Okay, so this is the concept of phototropism. Apart from that, Oxine is very much helpful in apical dominance. So what do we mean by the concept of apical dominance? A plant grows in two ways. Consider if this is the plant, one is increase in the length of plant. Okay. And another is by increase in the branching, the width of the plant is increasing. Not width of the stem. I'm talking about the width of the plant. You know, you must have noticed that shrubs are having branching, no? More branching, more bushy the shrub is. So same way, Oxine is promoting the apical dominance. Apex means tip. So The main function of auxin is, as it is stored in the root tip or shoot tip, it is making sure that the plant is increasing its length. Okay, so what it does, two types of buds are there present in the plant. One which is present at the apex or the tip is known as apical bud, and which is present at the branches. You can see here in this diagram, see these are the lateral buds at the base of branches. So if they grow, the new branches will be produced and the plant will have bushy appearance. That is called lateral dominance. What auxin does? Auxin increase the growth of apical bud and inhibit the lateral buds growth. So that way it is making sure that apical dominance is there. So obviously, हाँ वही तो हुआ. Apical will grow and lateral side will not grow that much. That is what is the dominance one. So you know there is a process called decapitation. आपने देखा होगा बहुत बार गार्डन्स या ये सब जगह पे वो बड़ी सी कैंची लेके वो छांट देते हैं प्लांट्स का जो टिप है. वो क्यों छांटते हैं? So that the plant will have more branching and it can become bushy. Especially for teas. Tea plant they do it because more branches means more leaf, more tea. So that's why they do it. This process is called decapitation. Okay. So next plant, gibberellin, plant hormone that is gibberellins. They are also called gibberellic acids because their nature is acidic. So E. Kurosawa actually observed one disease which is known as Bakane disease or foolish seedling disease. What is this foolish seedling disease? Can you see this photograph presented in the PPT? You can see the Bakane, after Bakane infection in the rice field, he observed that the normal rice is marked as P, which is not having proper growth in the field. Once the E. Kurosawa actually sprayed the field with the extract of Gibberella fujikuroi fungus, he observed this kind of growth that is demarcated by A. So can you see the difference? Don't you think it is foolish difference? Why foolish? Because it is astonishing. छोटे छोटे राइस प्लांट जो देखने में ऐसा लग रहा था कि जिंदा ही नहीं है, they grown up to the level of big height. Understood? So that is what is the chamatkar of gibberellin. So they observed this and then they said that the active ingredient that is extracted or that is responsible for this growth is gibberellic acid. And auxins are limited, but gibberellic acids can be more than 100 types. And as per their discovery, they are marked as GA1, GA2, GA3. So they are present in fungi, they are present in higher plants. So aap socho, if this Gibberella fujikuroi is infecting a plant, that is boon for that plant, right? It will have big height. So this kind of thing happened. That's why this disease was foolish seedling. Why E. Kurosawa extracted substance from Gibberella fujikuroi? Because क्योंकि उन्होंने देखा था कि एक फील्ड है उसमें अचानक से कुछ प्लांट्स है जो बहुत लंबे, while others are very small. So he wanted to know what happened. Then he observed that people used to say these are foolish things. That's why they grow so much. But he wanted to see what is the reason of this foolishness. So he observed that the fungus is responsible for such foolishness. And that's why he extracted the extract of fungus and sprayed it into plants which are having stunted growth. Stunted growth means they don't have that much high growth, and observed the miracle. Okay, so GA3, that is a type of terpene. Terpene is the chemical nature. Yes, Rutu want to ask some question. Up to a certain extent, yes, अच्छी बात. But अब हम उस पे डिपेंडेंट ही नहीं है कि डिजीज हो. हमें पता है जिबरेला फुजिकोरो एक्सट्रैक्ट में क्या होता है. We are producing it in the lab and spraying to the plant. कौन? फंगस कुछ नहीं करता है. फंगस के अंदर जो केमिकल है that does it, no? वो इन्फेक्ट कर रहा है ना. इन्फेक्ट कर रहा है मतलब इट इज अ पैरासाइट. इट विल it may have other harmful effect, but it is not causing harm to the plant that way. Okay, so GA3 is a terpene, and it is most studied gibberellic acid. So we will look into the functions of gibberellins. So we will talk now about the functions of gibberellins. The one function is very clear to you, and you can see the many examples. Can you see this gentleman holding grapes in his hand? You can see the enlargement of grapes. You can see the enlargement of apple, and then in case of cabbage also, the yes, the variation is remarkable. So obviously they increase the length of axis and they lengthen the grape stalk. What will happen if the stalk of the grape is long, then more number of grapes can be there. So it is very profitable for the farmer. It can be having improved shape in fruit like apple. Gibberellin is a very good fruit to very good hormone to delay senescence. That means it is not allowing the plant to grow old. Now plants generally don't grow old quickly, but the thing is that the main problem, once the fruit are ripened, there is a general tendency of fruit to get detached from the plant, and once they are detached, we don't have much choice to maintain their shelf life. What happens generally, जैसे ही वो plant से अलग हो जाते हैं, फिर उनको हम ज़्यादा दिनों तक रख नहीं सकते. They get destroyed. So what gibberellin can do is they can keep the fruits in the plant for longer time. They delay the senescence. Now, what do we mean by senescence? In case of plant, the aging is synonym to the senescence. Okay. GA3 is used to speed up the malting in brewing industry. What do we mean by malting process? Malting process means, want to say something? So the process of utilizing malt to produce the alcohol, the process is accelerated by GA. So there can be increased length in case of sugar cane, and for sugar cane, if the stem size is increasing, that is good. Why? Because we are taking the stem only as a product. Apart from that, if we are spraying gibberellin in young conifers, what are conifers? Cone-shaped plants. Okay, it is quickening the maturity period, means they quickly get matured and they have early seed production. So how it is beneficial? Apart from that, we get many types of products from these plants. So turpentine oil, we get from turpentine. Then sago palm is a conifer. So if they are getting matured early, we can expect these products early. No, that's what the benefit. Okay. Then apart from that, it promotes bolting. So what is this bolting? Here, just before flowering, the internodes elongate. So what will happen? If the internodes elongate, overall size of the plant will do what? It will also increase. I guess everyone know what is internode. Riva, you know what is internode? Sujal? Priyanshi? Krishang is having one question. I don't allow any internal talking in the class. Yes. There can be, depending on specific type of fruit, at the time of production, hormonal sprays are done. Those are hormones only. Definitely. But you are not eating it from surface, no? You are cleaning the fruit and eating. नहीं, तुम्हें कुछ नहीं हो. I taught you the whole chapter number 22, which says that hormones need receptor to work. After that, asking this question is. So we are coming to the third hormone, that is cytokinin. Cytokinin is a very important hormone because it is extensively used in tissue culture to produce callus, that is undifferentiated mass. How callus is formed? If a plant is going for de-differentiation, we studied yesterday what is de-differentiation, then callus is produced. So F. Skoog, he is one of the pioneer in plant tissue culture, and MS medium, that is Murashige Skoog medium. He is discoverer of Murashige Skoog medium or MS medium. It is used, it is a medium which is very much used for plant tissue culture. So what he observed that in case of internodal segment of tobacco stem, the callus is formed only if the medium was supplemented with yeast extract or coconut milk or some DNA. So they were under research that what is present here, which is promoting callus formation. That means simple MS medium will not promote callus growth, but if you are adding coconut milk into it, that will produce callus. So that means something present in coconut milk is responsible for callus formation. So they went for chemical research of this substance and identified the active substance, which is known as kinetin. So kinetin is furfuryl aminopurine, very cute name it is having. And it is an adenine derivative, and it can be easily obtained from herring sperm DNA. Herring is a fish. Herring fish sperm's DNA, it can be obtained. But kinetin is not found in plants naturally. You have to, if you want to promote callus formation, you have to give it to plants. Second one is zeatin. It is natural substance with kinetin, which is present in plants. And zeatin is giving kinetin-like activity. And natural cytokinins are generally synthesized in the areas like shoot buds, young fruits, and they are giving various functions. First of all is cytokinesis. Its name is cytokinin because it is promoting cytokinesis. Second thing is it is helpful for production of new leaves, helpful for increase in number of chloroplast in the cell. And next important thing is it is promoting lateral dominance. That means if you want to make a plant bushy, then you need to spray cytokinin. So the balance of cytokinin and auxin decides the growth of the plant. Okay. So if a plant is having apical dominance, that it is just elongated but getting very thin, in that case you can spray cytokinin to give it rid of apical dominance. Okay. So it is actually responsible to delay the senescence. So you found out that all the hormones are which are promoting hormone are delaying the senescence. Okay. Now we will talk about the opposite type of growth regulator, that is growth inhibitors. So first one of them is ethylene. Ethylene is a gas. You studied a lot about it in organic chemistry. Ethylene, C2H4. So that is responsible for ripening of fruit. It is available in market in the form of carbide. Carbide. जिनके पास मैंगो या किसी चीज की खेती है उनको पूछना what they do after harvesting the raw mango or the fruit. They put them into the jute bags, that is gunny bags, put carbide into it and keep it for some time. If you don't want to do it, put some potatoes. It will get ripened. They release it and they grow. Okay. So it is responsible for aging of the plant. So aging means growing. So it is causing depletion of or decrease in the number of chlorophyll, increase in the number of xanthophyll, carotene, so that the color of fruit will change. But let me tell you, it's not prescribed to eat fruits which are ripened by using ethylene. There is a very important thing. There is nothing harm, but it's not as tasty as the one which is ripened in the tree. Why? Because many a times why they treat the raw fruits with ethylene, they put ethylene as a position at a specific position. So what will happen? The one side of the fruit is getting in exposure of ethylene, the other one is not. Many a time after getting mango, आपको ऐसा दिखेगा कि एक वाला साइड मीठा है, एक वाला साइड खट्टा है, और एक पक गया है, दूसरा नहीं पका है. So you will understand that these are the mangoes which are not ripened in the tree. वो झड़ गए होंगे or they got detached from the plant and like I don't know, in Ahmedabad it is very difficult for you people to see a tree or own a tree. Own a tree. I belong to a small place, same old Kalyani, where I have a house, and surrounding house I have mango trees, supari tree, banana, papaya, and gardens and all, and we can get, it's not a farmhouse, don't worry, but still we have that facility. So in some cases you can clearly demarcate, okay, this is the fruit that is present in the tree and in ripened condition it is harvested. But nowadays we are in hurry, we don't wait for such things. Okay. So what are the functions of ethylene? First of all, it is promoting the horizontal growth of seedling. Horizontal growth of seedling means it will increase the thickness of the seedling. Okay. Apart from that, it will help in the apical hook formation. You can see the apical hook, the apex portion bent to form apical hook structure. You can see in the PPT. Apart from that, it promotes उससे ये होगा कि ये जो सीड है ये जमीन के नीचे से बाहर आता है. So this yellow part is having your cotyledon. So that's why. So apart from that, as I said, that it is contacting abscisic acid and promote exercisation or senescence. It is promote ripening of fruit and break the dormancy of bud dormancy or seed dormancy. Matlab, if ethylene is there, it is always in a hurry of making everyone old. So if you are providing it to seed को भी वो बोलेगा उठ चल फटाफट जर्मिनेट हो, बट को बोलेगा चल फटाफट फ्लावर बन. Again, do you have receptors of ethylene, Rutu? Rutu is so much not at all happy with the animal hormones. She is interested to inculcate plant hormones. So you don't have growth hormone in you? I told students to connect the plant and animal chapter at the beginning of this. And students are taking it too seriously. Kindly don't take. So other functions involve rapid internode or petiole elongation. What is petiole? The other functions of ethylene, as I said, was petiole elongation. So it is observed in case of rice. For rice plantation, the base of the rice plant, that is root and nearby area, should be immersed in soil, water. That's why rice is generally cultivated into the area with more water supply. In dry area, it is not cultivated much. Okay, but this ethylene makes sure that the internode or petiole portion of the rice will get elongated so that the panicle, the rice, main part of the rice, the grains, they will should always stay above the water, because in presence of water that will get destroyed, and the base of the rice plant, that is the root, should be immersed in the water. Apart from that, it is promoting the root growth or root hair formation. Can you look into this image how vigorous root formation, cotton-like structure is observed in this case? It is inducing flowering for pineapples and flowering in mango. That's why many a times if mango plants are not having initiation of flowering in the correct season, people spray ethylene to induce flowering, because from these flowers only later on the fruits will be formed. So that's why ethylene is used in agriculture. The most widely used source of ethylene is ethanol, and it is aqueous solution, so comparatively very easy for us to use it because you can spray it. Ethanol also hasten the fruit ripening of tomato. You can see in this plant where ethylene is sprayed, the color of tomato is changed. Other one is green in color. Actually, in some cases, it can demarcate the formation of flower. For example, in case in cucumbers, if ethylene is sprayed, it promotes the formation of female flowers. So, what is the benefit? If more female flowers means more fruit, so it will be more harvest. So these are the different ways these characteristics of were identified, and they were utilized by agriculture specialists. Now we will talk about abscisic acid. Abscisic acid was discovered in mid 1960s, and it is one of the major inhibitor hormone. Actually, they are carotenoid derivatives. You must be knowing, grade 12 student must have studied, grade 11 will study in future. Carrots are rich in carotene. That's why they are called carrot. Their scientific name of, you also have that. Doctus carota is the scientific name of carrot. Okay, so it regulates the abscissization and dormancy. So, what do you think? Should plants be dormant or they should not? Is dormancy required by plant or they should not be dormant? See, before germination, every seed take a pause. Every seed go for dormancy for some time. You must, I don't know if you are aware, like these are the chapters I very comfortably teach in like small places, because many students are coming from farmer background. They know after harvesting rice, same year they don't plant that rice immediately. They have to keep store that seed so that that seed can get matured, and then they can use it for plantation in the next year. Okay, so that's why dormancy is in some cases good, but the thing is that in some cases the seeds do not come out of that dormancy. So the intricate role play or the level of hormones decide that seed should come out of dormancy or not. Okay, so abscisic acid is something which is acting as a inhibitor of plant growth or mechanism. It is inhibiting germination of seed. All its factors are inhibiting. It is promoting the closure of stomata, which is not at all good. Because no gaseal exchange means no photosynthesis, no respiration. Okay, but what is the benefit of this? This is making the plant able to tolerate stress. That's why it is many a times known as stress hormone. So if plant is at water stress or temperature stress or have unwanted infection, in such cases, abscisic acid is very helpful to cope them up to the stress. It is helpful for seed development and maturation also. Obviously, now I wish to talk about the individualistic and synergistic role of PGR. That means what? We studied the individual function of each and every plant growth regulators, but that is not enough data. All of them are together decide how plant will grow. So there should be a synergistic growth. If too much auxin is secreted, cytokinin stops secretion, that is not good for plant. The perfect level of auxin, cytokinin, gibberellin, ethylene and abscisic acid is required. So that's why they should have a synergistic role or complementary role. Okay, so it will affect the dormancy in the seed or bud. It will promote the abscissization in the correct time. Abscissization at the correct time is equally important. Why? Because if fruit or leaf, old fruit or leaf is not falling, then new one will not be produced, and plant will not get rid of the unwanted excretory substance. Understood? Next thing is it is promoting flowering. It is promoting aging of the plant part. See, our aging and plants aging is not same. In our case, aging is faster. In case of plants, it is quite slow. Why? Because in our case, when we age, our whole body age. ऐसा नहीं है किडनी का एज हो रहा है हार्ट का नहीं है. Whole body together will get aged. But in plant, what happen? A part get aged, that part get detached, and plant is. So that's why they survive for more time. Okay. So they will promote vernalization. We will see what is vernalization. They will keep a balance between apical and lateral dominance, seed germination, plant movements. So obviously, whole of their role is very much important. So how these policies are policed? The way we have hormones, and hormones are controlled by pituitary, and pituitary is controlled by hypothalamus. In case of plants also, there should be some control. So these are known as extrinsic and intrinsic factors. Secretion of hormone and their function is either controlled by extrinsic factors, that means factors which are not part of plant. They are external factors like temperature, light. Understood? Whereas intrinsic factors are there which is controlled by the genome, that is DNA. That's why some plants are very healthy and some plants are not. That is decided by the genome. Okay. So obviously we cannot control the genomic factors because plants are born with that. But we can control the external factor, and that's what we are going to study next. That is role of light and temperature on flowering. So kindly look into textbook for this much portion and let me know if you have any doubt. So we will study about the role of light and temperature on flowering. First, we will talk about photoperiodism. Here, plants depend on the photoperiod. That is how much light they are getting and how much darkness they are getting. So depending on that, we classify plants into long day plants, which require more light period and less darkness. Okay. And there are short day plants which are requiring more darkness and less light. And day neutral plants have no correlation between the exposure of light duration. Doesn't matter to them. So obviously the long day plants are the plants which prefer to grow in the summer season. Because I guess you know that in winter the days are shorter and the nights are longer. So obviously, based on the light duration, we classify them like this. Now, how light photoperiodism works? Actually, the part of plant which can sense light and darkness is leaf, not flower. Okay, and it is thought that still no discovery or extraction of that hormone is possible, but there is a hypothetical hormone proposed by scientists which is known as florigen. The name of that hypothetical hormone is florigen, and it is said that it is migrating from leaf will sense the light and darkness, and it will send the florigen hormone from leaf to the shoot apex and induce the flowering. Because generally flowers are nothing but modification of apical bud or the lateral bud. So it is said that the leaf will sense the light and they will send the florigen over there. But जैसे कि हमने ऑक्सीन एक्सट्रैक्ट किया है, we know the chemical formula. ऐसे अभी तक फ्लोरिजन डिस्कवर नहीं हुआ. So you have scope to do something. So apart from that, I wish to talk about something very important, that is PR and PFR. For that I will just take some time. So before we go for vernalization, I wish to talk a bit about photoperiodism. So I am just noting down the points. I request everyone to pay attention. So the characteristics of photoperiodic responses, I am going to jot down here. So what are these characteristics? First thing is, obviously the photoperiodic responses are controlled by means of genetic. So genetic control is there. You know, this photoperiodism is such well sensed by plants that if consider it is a short day plant, or even if it is a long day plant, during the darkness, if the darkness is even slightly uh, what I can say, interrupted by sudden light, then also plants will not flower. So that means the darkness period they require should be complete darkness. It should not be interrupted by any kind of light. So photoperiodic responses are under control of genes. It is well explained that the photoperiodic responses are under control of genes, and it is possible that due to some kind of genetic manipulation, grade 12 will students will study this year, that how genes can be manipulated, and obviously by that we can yield varieties which can be having varied photo period. For example, NBRI, that is National Botanical Research Institute, Lucknow, is able to evolve a variety of chrysanthemum. Chrysanthemum is a flowering plant. Okay, and they were able to develop a plant. Chrysanthemum generally prefer to grow in winter season, but they were able to grow a chrysanthemum plant that can grow in the summer season also, because they were able to modify the photoperiodic requirement of that genetically modified plant. So what is the benefit? You don't have to wait for winter season. You can grow chrysanthemum in any season. Okay. Second factor that is very important is the critical day length. So what is this point of critical day length? So a specific, as I said, a specific period of day and a specific period of night is responsible for flowering. Now, if I'm talking about a plant like Xanthium, which is a short day plant, it will be requiring a specific hour of day, that is 15.5 hours. And if that plant is getting darkness hour, 15.5 means 24 minus 15.5 will be 8.5. So if it is getting even 0.5 hours less, that is half an hour less darkness, it will like fail to not grow, fail to flower. So long day, short day plant is related to flowering. Okay. So on the other hand, there is another plant which is called Heoschimus niger. It is a plant which require 11 and 13 days. That means 11 hours light and 13 hours darkness. So this shows that the division is always not very usual. And when the what I can say, when the after summer the winter comes, the length of the day slowly and steadily decrease. So all these plants are grown into a specific span. They are so critical that even if that span is gone, they cannot grow. Understood? So that is what is called critical day length. Okay. Now the third point, that is very important point, is inducive cycle. So what is this inducive cycle? Actually, what will happen if a plant is not getting the photo period? Consider I talked about Xanthium plant. It is required 8.5 hours of darkness and rest is light. So will they die if they don't get this photo period? No, they will grow. But that growth will be only vegetative growth. They will not reproduce. So for reproduction, they actually require this specific photo period. That means for Xanthium, even one day there is a situation that 15.5 hours daytime and 8.5 hours darkness is there. It will quickly induce the cycle of flowering, and later on it will start the flower. Understood? So this critical photo period is actually inducing factor for flowering. अगर ये नहीं आया, ऐसा नहीं plants मर जाएंगे, but they will not flower. And from our point of view, from the point of view of agriculture, obviously if a plant is not flowering, it is useless. Okay. Next is if we are talking about phytochrome. So what is phytochrome? We will talk about the role of phytochrome in flowering. So what are these phytochrome? These phytochromes are a kind of chemical. They can sense the light. Okay, so they can sense the two types of radiation. One is like red radiation, other is far red radiation. So I just wish to say how phytochromes are promoting flowering. If it is red, you know what is red color? Light. If plants are getting, it will inhibit flowering. But if red and then there is far red, then the flowering will be promoted. Again, if you give R, FR and R, again if you are giving it red, again it will be and if you are giving R, FR, R, again FR will be. So this is how phytochrome is responding in two ways. When phytochrome is exposed to the red light, it forms PR, which is a form which is inhibiting flowering. And phytochrome, when it is getting exposure to the far red light, it is forming TFR. Then it is the same phytochrome will be promoting the plant. So depending on wavelength of light it is getting. And last but not least, I wish to talk about the last hormone, that is, I told you there is a hypothetical hormone that's called florigen, although its activity is not described much. And this florigen is also responsible for induction of flowering. So can we move ahead towards vernalization? If you want to note it down, you can, or want to take a screenshot, you can. So now we will talk about vernalization. So the vernalization is applicable to some plants. That means they require a low temperature exposure before flowering. So normally plants, some plants blossom in the spring season. Why? Although the plants blossom or flower in the spring season. Why in the spring? Because before that they have winter. And that low temperature exposure before flowering is helpful for the induction of flowering. So, what is the advantage of this? During winter season, condition is not very much favorable for the plant. So, if plant is not flowering during that time, the during reproduction losses will be less, isn't it? बहुत ठंडी में पोलन कहाँ जाएगा, वो किसको fertilize करेगा? That will be a question mark. So this flowering period, the cold period, will make sure that during that period the flowering will not be there. It will be there after the cold period, that is after, during the spring season. है ना? So this is known as thing. Now there are some varieties which are called spring varieties. They are generally planted in the spring and came to flower and produce grains before the end of the growing season. And there are winter variety. आप जानते हो इनको बोलते हैं रबी क्रॉप, खरीफ क्रॉप. This must have studied. So winter varieties are planted in the spring, and they are, if these varieties, winter varieties, sorry, they cannot be planted in the spring. They are generally planted in the autumn season to harvest during the mid-summer. Okay, and there are biennial plants. Vernalization can be done with them artificially. They can be kept under cold temperature to have fruit or flower. Biennial are those plants which can survive for 2 weeks, what I can say. Second season, they don't survive up to the second season. In second season, they die. So they grow twice a year. मतलब 6 months उनकी life है. That's why we call them biennial. Okay, they're like sugar beet, cabbage, carrots. So in which season carrots grow? हम गाजर का हलवा सबसे ज्यादा कब खाते हैं? So cold treatment can be used to stimulate them photoperiodism. And with that, I am done with the chapter. Thanks for all your cooperation. We will study next chapter as mineral nutrition. So Monday next, I will first take your homework. Okay, so bye to all of you.